All right, so we're going to be picking up right where we left off. We left off last class talking about antibodies and their structures, how there's two identical light chains and two identical heavy chains, how each one has an antigen binding domain called the FAB region depicted here up on the board, and that there's an FC region that binds to antibody receptors called FC receptors. And they bind to the FC portion, the bottom part of the Y shape of the antibody, the FC portion of the antibody. Antibodies can be present in a couple different physical settings. They can be secreted and they can be secreted. We talk about B cells differentiate into plasma cells, and plasma cells are our professional antibody producing cell. So when these antibodies are secreted, oftentimes, most often, you'll find antibodies present within the blood. And so typically, if you're going to the doctor's office and they want to check your antibody titer levels, they'll do a blood draw and check your antibody levels there. When it's not free floating in the bloodstream, there's two non-free floating conformations for antibodies. So one is where it can be bound to the FC receptor on immune cells. Another is as the B cell receptor when it's not really bound to an FC receptor, but rather it's wedged within the plasma membrane. And so I've depicted that here within a plasma membrane. You see BCR. It's not bound to an antibody, but rather the B cell receptor is an antibody that has a hydrophobic region in the FC domain, and it's wedged within the plasma membrane there. So again, the key thing to note here is that we have two types of lymphocytes that kind of the focus here is we're going to be focusing on the foundation, foundational information of immunobiology. So when I say there's two types of lymphocytes, it can be a little more complicated than that because immunology can be frustratingly complex, but we're focusing on the foundational information here. So the two key types of lymphocytes, we have T cells and we have B cells. And again, the B cells constitute the main cell type within our humoral immune component in adaptive immunity. These are the cells that differentiate into plasma cells. Differentiate is becoming more specialized in their structure and function, and are the professional antibody secreting cells. And in addition to that, B cells are also one of our class of antigen presenting cells. I've listed again our list of antigen presenting cells on the board here. We have, and I've done this in the immunologist shorthand, so forgive me. We have macrophages, dendritic cells, and B cells are antigen presenting cells. That means they'll bind to antigen, digest it, and display it in small peptides on its surface. So here for B cells, what they'll do is they'll bind, they'll see free antigen floating around or present within its local microenvironment, bind to that free antigen, internalize it, and then break it into small peptides pieces and display it. T cells make up the main component of cell-mediated immunity and on the adaptive side. The main mechanism by which they carry out their function is direct cell-to-cell contact. They'll bind to other infected cells. They'll induce apoptosis. That's a programmed cell death. It's very focused and very targeted. Another form of cell death is perhaps like necroptosis, which happens in response to tissue injury, where it's really indiscriminate in how cells die that way. But apoptosis is very targeted, one cell at a time. And T cells also have a key role in modulating the immune system. We talked last class about gas and brakes. The immune system can't be all gas. If it was, that'd be a real problem. And so there's a flavor of T cells that help provide the brakes in our immune system to help regulate things. So as I pointed to earlier on the board here, each lymphocyte, both B cells and T cells, we talked about this last class as well, they each have a type of antigen receptor. For the B cell, they have the B cell receptor, and that is a membrane-bound immunoglobulin or antibody that binds the free antigen, and it's functionally an entire it has two FAB regions and an FC domain. And so it's going to look like functionally an antibody that is wedged within the plasma membrane of a B cell. T cells have a T cell receptor. That's what their antigen receptor is. It's going to resemble an antibody in some ways, but not entirely. It's going to more closely resemble the FAB domain of the antibody, and I've depicted the T cell receptor as TCR, but we typically don't refer to it as. We're going to talk about the specific nomenclature for the T cell receptor, but it functionally looks like a segment, the FAB domain of an antibody that also is wedged within the plasma membrane. What's key about the T cell receptor is it's going to be binding to peptide antigens that have been displayed to it. When we talk about antigen presenting cells, this is like asterisk here, this is key information. When we talk about antigen presenting cells, and we've been talking about they'll chew up peptides and they'll display that peptide somehow, but we haven't talked about like where that displaying goes to. They are displaying it to T cells on their T cell receptor. And so again, as I mentioned before, these share a structural similarity to immunoglobulins or antibodies. And like antibodies, they have incredible specificity. Last class, I talked about the antigen binding domain, as you can envision, is where the variability in antibodies and antigen receptors resides. And so for the T cell receptor, because it's functionally the antigen binding domain of an antibody, it has incredible variety and diversity of how many different kinds of T cell receptors we can possess that will then bind to very specific epitopes on antigens. And each T cell, kind of like with the B cell, each T cell has one and only one T cell receptor, but many copies of it littered across its surface. And so the key thing to note here is that T cells use the T cell receptor to bind and recognize antigens. And they only, and again, this is like the next key for asterisk here of another piece of the puzzle. We talked about how antigen presenting cells will display antigen, as an example, antigen presenting cells, when they display a peptide, they're displaying it to the T cell receptor. In general, T cell receptors can only see an antigen when it's been presented on a certain class of proteins that are called major histocompatibility class proteins. That's my last time saying that full phrase because we usually just refer to it as MHC. So I'm going to repeat that again with the abbreviation. T cell receptors are only able to bind and recognize their cognate antigen when it's been presented to them by MHC proteins. So when we look at the T cell receptor versus IgG, which is one of the flavors of antibodies we talked about, you can see when we look at their structure, there's a pretty near identical structure between the two of them. And again, that's because the T cell receptor is an immunoglobulin itself. However, we don't typically refer to the components of the T cell receptor the same way as we do with antibodies. With antibodies, we might talk about the light chain and the heavy chain. With the T cell receptor, we typically talk about alpha chain and beta chain. That's what's depicted here in blue and yellow. So major histocompatibility complex, or MHC, is broadly expressed across a variety of species because, again, it's a really key, important component of our immune system. In humans, we typically refer to as human leukocyte antigen, or HLA. But broadly, this class of protein and the genes that are associated with them, we typically refer to them as MHC, unless, again, you're talking about specifically human immunology. What's really important about MHC is that their main role is to display peptide antigen to the T cell receptor. And so what's important to note is that there's two flavors of MHC. I talked last class, and I think the class before, about each nucleated cell in our body has this almost like ID protein on the surface that kind of identifies them as our cells. that is one of the flavors of MHC. That's called MHC class 1. So MHC class 1 is unique to you. You inherit both copies from your parents, and it is expressed on the surface of every nucleated cell in your body. So every cell except for red blood cells. The other flavor of MHC is called MHC class 2. That's only expressed on antigen-presenting cells. So that means antigen-presenting cells, when they're displaying peptide antigen to the T cell receptor, they're doing it using MHC class 2. This is oftentimes a really promiscuous process, meaning there's a constant dynamic of surveillance and displaying. So antigen-presenting cells are constantly engulfing antigen-presenting cells, particularly macrophages and dendritic cells. I'll give that caveat because B cells have a very specific B cell receptor. Again, innate immune cells are fast but dumb. Macrophage and dendritic cells are constantly engulfing, digesting, and displaying peptide on MHC class 2. With MHC class 1, our cells are constantly displaying what is within them in case they might be infected or there might be some sort of issue internally with the cell, particularly as it might relate to cancer biology. And so MHC class 1 is going to be displaying a lot of internal components that come from within our cells, whereas on MHC class 2 antigen-presenting cells, they're engulfing things outside of themselves to display what they've eaten. And this is, again, really promiscuous, and they'll be displaying any peptide that they've either engulfed for antigen-presenting cells on MHC class 2, or anything that's being internally processed with our normal healthy cells on MHC class 1. When we look at the structure of MHC class 1 and MHC class 2, what they look like and what they can display is a little different. And so typically MHC class 1 can display peptides that are about 8 to 10 amino acids long, whereas MHC class 2 can display amino acids that are 18 to 20. And so for a small illustration, you can envision that in this binding pocket for MHC class 1, that there be a regular size hot dog displayed there, whereas on MHC class 2, based on its size, would be a Costco-sized hot dog. Costco hot dogs have a very special place in my heart, so I just wanted to introduce that. So why should we care about MHC class? Why should we care about MHC proteins? They are again, MHC class 1 is is a unique protein to you that you've inherited from your parents. And so it is the primary antigen. Antigen, again, is the immune response inducing material. It is the primary antigen that occurs in response to transplant rejection. And so that's when when you hear about tissue matching, MHC class 1 is at the heart of that, and oftentimes is what needs to be accommodated for during transplant surgery. MHCs are polygenic. That means there are a lot of genes that are associated with the formation of our MHC proteins. And each one is comprised of lots of individual specific MHC genes, particularly MHC class 1. MHCs are polymorphic. That means there can be lots of mutations that can emerge with MHC proteins. And so that means there's lots of different genetic variations that can occur that leads to the different copies that people can possess, even though we all have the same genes that are responsible for our unique MHC class proteins. In addition to that, they're also codominant. What that means is that MHC proteins, you express both copies of the genes that you have there, or you express both alleles or both copies of the genes associated with MHC class proteins that you inherit from your parents. And finally, it's surprising, the odds are surprisingly low of two people inheriting the same two copies of MHC class 1. So the only exception to that, as you can imagine, are people who are really close relatives, and oftentimes who are the ones that are organ donors for one another. Oftentimes, not exclusively, but most likely. I'm going to hit pause. Oh, here, I'll do one more slide and then we'll stop for questions. So what this looks like at a structural level is that we have our T cell receptor. Our up in the top left, and we have our MHC class 1 protein in yellow. And blue is just a supporting protein that stabilizes this interaction between the T cell receptor and MHC class 1. And in the middle, in between them, is an antigen that's being displayed by MHC class 1 to the T cell receptor. So I'm going to stop there and give space for a few questions. Yeah, I'll have a few questions at this point. Yes. Yes. Yeah, those are the flavors of antibodies that you'd find as the B cell receptor. And what I should say, I think I may have said this, but in case I didn't, what distinguishes those 5 flavors of antibodies typically is the FC domain. And so for IgG, IgM, IgA, D, and E, they all differ in their FC domain, which is why the receptors that identify them all are going to be binding at the FC domain are all going to be different from each other. Bless you. Yes. Yeah, so that's a great question. To my knowledge, they don't need to be perfect matches. Oftentimes there's a threshold. There's a threshold at which there's like a minimal likelihood of transplant rejection. What that threshold is, I don't know off the top of my head, but I'll actually make a note of that and try and see if I can find that out. So that's a really good question. Yeah. Yeah, that's a great question. So the way that's a really helpful and important distinction that I think bears repeating. B cells are antigen presenting cells, but they are not phagocytes. The way in which they're able to internalize their antigen, even if they're not a phagocyte, is that when that antigen binds to their B cell receptor, there will be receptor-mediated endocytosis. What that means is that they will internalize that antigen, not by phagocytosis, like engulfing and eating indiscriminately. With B cells, it's far more targeted to the antigen that only binds to their B cell receptor. Macrophage and dendritic cells, they will eat anything and everything that they can get their hands on, for lack of a better phrase. But the B cell will only internalize what binds to its B cell receptor. So it's a far more specific process, but still plays a critical role in our immune response. Thank you for asking that question. Yes. The antigen would bind to the FAB region. The AB stands for antigen binding. So the antigen binds to the FAB domain. FC, what happens here is that this binds to the antibody receptors on our immune cells, FC receptors. All right, I'm going to keep moving, but again, if you have a question, feel free to just shoot your hand in the air and I'd be happy to answer. So there are a family of proteins that help us distinguish and identify certain flavors of cells. It can be a bit of an alphanumeric soup. These family or class of proteins are called cluster differentiation. We usually refer to them as CD, and there's a number that comes after it. There are, as of now, about 300 and maybe 30 or 350 of them. Excuse me. Not going to ask you to memorize them, but rather we're going to focus on just 2 to 3 that are really important for our content for today. So T cells come in 2 flavors. Well, so they come in many flavors. 2 of the flavors that we're going to talk about right now are called CD4 positive and CD8 positive T cells. between and each one will determine what each one might their importance in what we've just been talking about is that they'll each display uh, their role is to display a certain class. Their role is to display a certain class of MHC to the T cell receptor during the immune response. The way this works is with CD4 positive T cells, what they're going to be primarily tasked with is going to be on MHC is seeing what's displayed on MHC class 2 by antigen presenting cells and binding to it with its T cell receptor. And the reason why this is unique to CD4 positive T cells is that the actual CD4 protein that's on the surface of these cells, is depicted here on the slide, it actually helps to stabilize this interaction. And so the CD4 protein is really critical for the ability of antigen presenting cells to find the T cell receptor with the corresponding antigen receptor. What's really unique about these cells is that this marker, excuse me, I do not have enough water today. My throat's getting itchy. What's really unique is that in HIV infection, CD4 is actually what is leveraged to enter into cells, and so that's why every virus specifically targets something. So the HIV virus specifically targets CD4 positive cells, CD4 positive cells, which are oftentimes T cells. And so that's why there's a really severe immunodeficiency there, because it's obliterating a really key cell population within your immune system. So again, CD4 protein on the surface of T cells, so we refer to them as CD4 positive T cells, it stabilizes MHC class 2 presenting to the T cell receptor. In CD8, its role is to stabilize MHC class 1 displaying to the T cell receptor. And so it can be a little hard to remember what MHC is stabilized by what CD protein on T cells. So a trick that helped me when I was first learning this is you can think of this as like it's always a product of 8. And what I mean by that is CD4 binds to MHC class 2. 4 times 2 is 8. CD8 stabilizes MHC1. 8 times 1 is 8. So, and it's just like a helpful trick to try and remember what CD protein on T cells corresponds to what MHC class protein. It's always a product of 8 whenever you multiply those together. But before you know it, you guys will just memorize this down cold. So you might not need that very soon. The flavor of T cells that typically are CD8 positive, we usually refer to them as cytotoxic T cells. And you'll see why momentarily. But what this looks like depicted here is that we have our T cell receptor, we have our MHC class 2 protein loaded with a peptide antigen, and we have CD4 to the right in blue present to help stabilize this interaction. I'm going to do 2 more slides, and I'm going to give space for a couple questions here. So what this looks like is we'll first focus on one pathway, and we'll focus on the other pathway, looking at CD4 and CD8 positive T cells, and kind of like what this picture looks like. I summarized this really briefly earlier in the lecture, but just to repeat it again, for antigen presenting cells, they're the ones that display and express MHC class 2. Because of them displaying, they're typically going to be displaying something that doesn't originate from our cells. It's usually some sort of invading pathogen or something that is in the external environment. So because of that, we call that the exogenous pathway. Whatever the antigen is that it's displaying originated from outside itself. So they're going to either digest by phagocytosis or receptor-mediated endocytosis if it's a B cell. And it's going to enter into the lysosome or the phagosome by phagocytosis. But the main picture here is that they're going to be digesting and loading that peptide, excuse me, on an MHC class 2. MHC class 2 will move to the membrane and display it to the T cell receptor, and that interaction is stabilized by CD4. So again, this is going to be originating from outside the cell. It's going to be engulfing it, endocytosing it, digesting it, loading on an MHC class 2, and displaying it on the surface to the T cell receptor that's stabilized by CD4. This is going to then trigger the corresponding T cell to be activated, to proliferate, and to secrete a variety of soluble proteins and cytokines that mediate the downstream immune response. For the endogenous pathway, what this means is that whatever is going to be displayed originates from within the cell. So if this is an infected cell, a virally infected cell, it might be able to, there's this constant interrogation of what's going on by what's being produced or translated, what proteins are being produced. And so the way in which this works is that either the viral protein that is being produced because it's hijacking our machinery, or self-proteins in case of a cell that is healthy and being assessed. It's a key organelle in our protein synthesis process is the endoplasmic reticulum, or the ER. So what's going to end up happening is that this protein is going to be pumped back into the ER. And this is going against a gradient. So whenever you're going against a gradient, you need energy to do this. This is going to be using the ABC transporter, which is an ATP transporter. So this is going to be an active transport process. Whatever's being produced is going to be pumped back into the ER and loaded on the MHC class 1 and displayed for a CD8 positive T cell to interact with on its T cell receptor, if it in fact is seeing its cognate antigen. So the whole idea here is that it's a way of interrogating all of the proteins that are being produced. Because typically viral infection will hijack our machinery to replicate itself. And when that happens, there's typically going to be pieces of that protein produced. In the case of coronavirus, spike protein is a key antigen here. And so when your upper respiratory epithelium is infected and producing lots of spike protein, in the case of a coronavirus SARS-CoV-2 infection, those cells might be detected if in fact spike protein is then displayed on MHC class 1, because again, it's just taking what's being produced and displaying on the surface and waiting for a cytotoxic T cell or CD8 positive T cell to identify it and release cytotoxins as displayed here to kill the infected cell. In the case of a healthy cell, if it's displaying something that originates from us, like cytoskeletal proteins, that should not elicit an immune response. So when the T cell receptor sees this antigen, if it's cytoskeletal proteins, there's no immune response. It is a problem if cytoskeletal proteins elicit an immune response. And we'll actually talk a little bit later about some examples of issues like that and how it's dealt with, that's programmed into our immune system. I'm going to stop there and give space for a couple of questions. Yes.
Could you explain the function of the transfer?
Yeah, so whenever proteins are being synthesized in our cells, part of their pathways to travel through, it's called the ER, the endoplasmic reticulum, and it's usually flowing in one direction. So there's a gradient where it's going from, it's moving away from a couple organelles out towards our plasma membrane to secrete these proteins usually. What this process is doing is trying to work against that gradient. So it needs energy to do that, to work against the gradient. And so the ABC transporter is an ATP mediated transporter, which means it uses ATP, it uses energy to pump against that gradient so that the protein that's being produced can be taken back up into where it can be loaded in the ER on MHC class 1, because our cells are also making MHC class 1. It's a protein. We have genes that encode for it, and so we make it. So when MHC class 1 is made, some of the protein that is able to be pumped up against the gradient can be loaded on MHC class 1 and then displayed on the surface. You guys look like you've been drinking from a fire hose a little bit. If something doesn't make sense, that's on me, not on you. So if you feel like something is confusing, that's not your fault. That's my fault. Yes. Yeah, we're going to talk a little bit more about that later on the lecture. But yeah, the CD8 positive T cells are called cytotoxic T cells because, as what's displayed here, they're going to when they find their cognate antigen, the antigen that they're destined to respond to, they'll release cytotoxins to kill that infected cell. That's why they're called cytotoxic. TH referred to as a helper T cell. I have an entire slide on that. I should have changed the labeling there, so that's on me. But yeah, it's called a helper T cell, and we're going to talk about those two cells in a few slides. Yes. This diagram right here? Yeah, so this is just depicting at a structural level. So the take-home message for this slide is just to see what it looks like where the T cell receptor with interacting with MHC class 2, displaying the cognate antigen for that T cell receptor, the antigen that this T cell receptor is destined to respond to, and that this interaction is mediated by CD4. What is hard to appreciate here is that, so if the T cell receptor is up top here, that means the T cell would be up here, and the antigen presenting cell will be down here. CD4 comes from the T cell, so this is a membrane-bound protein on T cells, on certain T cells. And it actually will come in and actually interact with MHC class 2 to stabilize this interaction. These are great questions, by the way. And again, I'll say if something isn't making sense, that's on me. And if you have a question that's kind of burning in your mind, it's possible that someone else has the same question. So again, really want to encourage as much as possible for you guys to ask any questions that are kind of bouncing around your mind at all. All right, we'll keep moving. And again, if you have a question, shoot your hand up in the air. Some important key facts here are that we talked about how each antigen receptor recognizes one and only one antigen, or one and only one epitope. Largely true. Again, immunology is a little complex. Oftentimes, what it comes down to is that we'll bind and only bind to structurally related epitopes. As an example, it's just kind of an easy example to keep going to. When we think about the coronavirus and the different strains that emerged and efficacy of vaccines, there were the different variants that emerged. Off the top of my head, I feel like I can remember Delta and Omicron, and then a bunch of other ones I'm forgetting right now at the moment. But those were changes that happened to specific antigens on the coronavirus, particularly spike protein. And so what ends up happening is that the question became, will the vaccine still recognize and will it still promote the immune response to appropriately recognize this new variant? And so in a lot of cases, that was the case. And because they were all still structurally related, there might have been very small polymorphisms or mutations that happened, but that were still able to be recognized by the antibodies that were produced or by our T cell receptors. And that's largely because we talked about how MHC class 1 can bind to 8 to 10 amino acids. One change in amino acid, it can still bind to it, but the affinity or the likelihood of that binding might go down. And so the idea is that it can still bind, but it might not bind as well. And so you can also think of this as a lock and key mechanism where when you're putting whenever a lock has been, or not a lock, whenever a key has been like slightly altered, it doesn't go in as well, but it can still sometimes work depending on the degree of change that happens. But what's important to note is that for B cells and T cells, they start off naive and they'll get differentiated. And once that differentiation happens, again, that's a specialization in structure and function. Once that happens, they're going to be producing that one and only one specific antigen receptor forever. And so they are destined to try and look for and bind to the corresponding antigen to their antigen receptor, or the cognate antigen that they're destined to interact with. And again, this explains why there's such a specificity in our immune system that each lymphocyte has one specific antigen receptor and that binds to a very specific epitope. But what's important is that despite us having only one lymphocyte at first during development for each epitope, we need many lymphocytes to produce an actual uh, uh, robust immune response that is needed for the diversity. And so we'll actually talk about what that looks like in response to an antigen. And so we're going to take a moment to describe the origin of our lymphocytes and then the differentiation of how we get this diversity of our different lymphocytes. And so there's going to be some key locations that we talk about that describe how our different lymphocytes emerge and where they emerge and where they go to in maturity. So we talked about hematopoiesis. We'd had that slide that had like the lineage of all the immune cells that originate from one stem cell, the hematopoietic stem cell. So that is the main stem cell that originates from our bone marrow. So typically the hematopoietic stem cell in the bone marrow will differentiate into one of two options, the common lymphoid progenitor, CLP, or the common myeloid progenitor. Because we're focusing on adaptive immunity, we're going to focus on the common lymphoid progenitor. So what ends up happening here is now and I'm not asking you to clarify here. I'm not asking you to memorize this lineage, but what I want you to focus on is the location that cells emerge. And I think it'll be clear, like, and I'll highlight what cells you need to know and what locations. So B cells from the lymphoid progenitor, B cells will, the common lymphoid progenitor will differentiate into a B cell in the bone marrow. That's why it's called a B cell, because it originates in the bone marrow. For T cells, that lymphoid progenitor will actually leave and go to the thymus, and T cells will, they'll differentiate into a T cell in the thymus. They're called a T cell because they are originating in the thymus. After that, typically, and what I should say, I'll pause here, the bone marrow and the thymus, because that's where B cells and T cells originate from, we typically refer to those two places as our primary lymphoid organs or primary lymphoid tissues, because that's where we get that's where these cells mature and originate from. From there, they'll actually leave their primary lymphoid organs and go to secondary lymphoid organs. And the most common one that is littered across your body is the lymph node. And so lymph nodes are the most common and popular secondary lymphoid structure. The next two are the appendix and your tonsils. Because they're secondary, you can lose those structures, like some people have lost their appendix or had their appendix removed or had their tonsils removed, and that doesn't affect your ability to generate an immune response. If you lost your thymus, that would be a huge problem, particularly early on in age, because that's where you're generating these T cells that are key for the whole library of T cells that your body's going to produce that can respond to any antigen. And again, it's not important for you to memorize the order of operations in terms of the lineage, but rather where B cells and T cells originate from and what are the primary and secondary lymphoid organs or structures. Yes. The thymus is right in your thoracic cavity. Okay. So there are two means by which differentiation can occur in lymphocytes. We can have antigen-independent differentiation, which means differentiation is happening in the absence of antigen. And then we'll have antigen-dependent differentiation, which means it's happening in the presence of it and requires it. So we have our hematopoietic stem cells and our lymphoid progenitor that these lymphocytes are originating from. These stem cells, or these progenitor cells, which means the less mature version of our cells, these stem cells are going to give rise to antigenically committed lymphocytes. What that means is they're going to give rise to lymphocytes that are committed to respond to very specific epitopes on antigens. So this is happening during development. And each of these cells, again, makes one and only one antigen receptor. Their destiny is to see that one specific epitope on that that the antigen receptor recognizes. And this is all happening during development. This is all happening before birth, where as your immune system is developing, you are generating a library of lymphocytes with a library of antigen receptors. The generation of this library of lymphocytes, when they're naive meaning not differentiated or not activated, when they're activated, they'll undergo differentiation process and they're not and they're prepared to be more functional. Whereas naive, they're unactivated and not quite functioning quite yet. The generation of this library of naive lymphocytes, again, happens during birth. And all of this is happening, this differentiation to create these antigen-specific lymphocytes is all happening without antigen and happening before birth. Now, there's been, I think during last class, and I think one question as well earlier today, seemed to touch on a really important process where if we're generating so many different kinds of antigen receptors, if it's trillions of possible antigen receptors, how are we careful to make sure that only the antigen receptors that don't recognize self as foreign are eliminated? How do we select if it's if it's leveraging the the MHC gene or the MHC gene, sorry, the antigen receptor genes that help create such a diversity, if it's going to eventually create an antigen receptor that might recognize perhaps collagen or tubulin as foreign, how do we get rid of those? And so one of the ways that happens, one of the key ways that happens is via a process called positive and negative selection here. So first, positive selection is only going to be it will select for the T cell receptors that appropriately, sufficiently enough, bind to MHCs. For CD4, it's going to be MHC class 2. For CD8s, it's going to be MHC class 1. So the T cell receptors that adequately, again, bind to MHC on these thymus self cells. So these are cells within the thymus during development that are uniquely tasked with testing these new emerging lymphocytes to make sure that they are, for lack of a better phrase, passing a quality check. Excuse me. If they do not adequately bind to MHC, which means they wouldn't be promoting a sufficient response in response to antigen, those cells are going to undergo apoptosis. They're going to undergo programmed cell death and not be part of the final immune response because they're not doing a good enough job of interacting with MHC to induce an immune response. For the negative selection, apoptosis will occur in some of the cells that bind too well to our self cells. So what that means is these thymus self cells during development are really good at displaying self antigen to check for T cells that recognize self as foreign. So during this negative selection, if these T cells that have passed the first quality check are binding too well to MHC, what that means is, there's like an appropriate range of interactions. If it's not binding well enough, they're not going to induce enough immune response. If they're binding super well, that means they might bind when there's not the right antigen there, and it might promote autoimmunity. And so they'll get rid of those T cells during this negative selection process. And so what's going to end up happening is that because there's such a large number of possible antigen receptors, and the same process happens for B cells as well, at the end, about less than 5% of the cells at the end are going to be what remain. Because again, there's so many proteins that our body expresses that are part of self that are going to be QC'd out during negative selection, and a lot of cells that might not be appropriately interacting during the positive selection that are eliminated. So in the end, it's about less than 5% that pass this selection process. When we have these lymphocytes that pass this process, and then during post-gestationally, when an antigen is encountered, what's going to end up happening is that when the specific lymphocytes that have the antigen receptor that recognize a unique epitope on the antigen, it's going to become activated, and then it's going to also massively proliferate. So the best way to illustrate this is that whenever you've gotten a cold, you may have noticed that your lymph nodes might be really tender or might be really big or might be really frustrating as well. That's because in the lymph node, your T cells are this proliferation is an incredible degree of proliferation. And that's what's happening. You have your T cells and your B cells that recognize that antigen expanding massively to an extent where once they reach a point of proliferating sufficiently, they'll then go to the site of infection to help eliminate the invading pathogen. And so the specific clone that corresponds to the antigen, again, will undergo massive proliferation to help fight the infection. And so specifically for antigen dependent differentiation here, once our lymphocytes via their antigen receptor bind to the specific antigen, it's going to promote its activation. It's going to massively proliferate, and it's going to also differentiate into what we call a fully active effector cell. Effector cells in our immune system, this is kind of like a catch-all term, but it's the cell that mediates the immune response. There's going to be lots of flavors there's going to be a few flavors of T cells, T cell effector cells that we talk about. But the main thing to take away is that effector cells are the ones that mediate the immune response. As we talked before, for B cells, they get activated and mature into a plasma cell. And the plasma cell is the main effector cell that produces massive amounts of antibodies. For T cells, there's lots of flavors that are of effector cells that have emerged, and some that are still being identified. But there's going to be three main flavors that I want to draw your attention to that we've already referenced a little bit today. We have our cytotoxic T cells. These are the cells that are tasked with killing our self cells that seem to be infected with some sort of foreign pathogen. This is the CD8 positive. And so we usually say CD8 positive or cytotoxic T cells. The next class is our helper cells. These are our CD4 positive T cells. Their task is to try and go to the site of infection or the site where this invading pathogen is, and to equip the other cells to fight the infection or fight the infighting pathogen. And so we typically will refer to these as either helper T cells or CD4 positive T cells. And finally, I talked about gas versus brakes, that there's a cell population or a flavor of T cells that are the brakes. These are our regulatory T cells, or we usually refer to them as Tregs. They are typically tasked with helping to try and suppress the immune response so that it's not always firing, but rather helping to regulate its activation and its function. I think I'm going to hit pause there and give space for a couple questions. Yeah, go ahead. What do they do? Yeah, that's a great question. So the primary lymphoid, I'm glad you asked that. The function of primary lymphoid organs is where our lymphocytes mature and become a B cell or T cell. Secondary lymphoid structures or secondary lymphoid organs are the sites where our T cells and B cells will typically see antigen, where it's like you can almost think of our lymph nodes as almost like small immune centers of like immune activity, for lack of a better phrase. Because what will end up happening is our lymph nodes are all connected to each other and connected to various parts of our body through something called the lymphatics. If there's free antigen somewhere in our body, it'll drain to the local lymph node, or the antigen will be carried by a dendritic cell or macrophage to a local lymph node and try and find the corresponding T cell that recognizes that antigen. So it's at the lymph node where this immune response is happening, where antigen presentation is typically happening. Thank you for asking that. I should have clarified that. Yes. Oh yeah, I should have touched on that. So tolerance typically can be defined as promoting our immune system to tolerate some sort of antigen. For example, one dynamic of our immune system is sometimes things are identified as dangerous that are not. Pollen, cat dander, peanut, for example. And so the idea is can we try and promote tolerance when our immune system in its current state is as seeing this as antigenic or seeing it as immunogenic, promoting an immune response. So for an example, there's been a lot of work identifying what are ways we can promote tolerance to certain food allergens, like peanut, for example. And so regulatory T cells or Tregs play an important role in promoting oral tolerance in food allergy. All right, I'm gonna try and see if we can press forward and finish today. I think we can do it. Okay. The activation of B cells, we've kind of referred to this a few times, but I think it'll be helpful to try and depict this really quickly. Typically, B cells will bind to free antigen. T cells rely on their cognate antigen, the antigen that they're like destined to try and respond to. They wait for it to be presented to them, whether it's through MHC class 1 for a CD8 positive T cell or MHC class 2 for a CD4 positive T cell. B cells are different. They will bind a free floating antigen. And when that happens, when the free floating antigen binds to the B cell receptor, it'll be endocytosed, and that'll trigger the activation of that B cell. And so what will end up happening is that the B cell will rapidly proliferate, meaning it'll expand and grow and double and triple and so forth, and it'll differentiate into a plasma cell. What's also important to note is that there's a flavor of B cells that helps promote long-lasting memory called memory cells. These are long-lived versions of B cells that emerge in response to this antigen and that reside in our secondary lymphoid structures for responding to subsequent exposures. And so what this looks like, the reason why this is so important that we have these memory cells is because typically you can envision if the very first antigen you're ever seeing after birth, you have one lymphocyte that has the antigen receptor for that cell. When that T cell sees its cognate antigen, it's going to proliferate and expand. A subset of those cells will become memory T cells that will stick around and reside for subsequent exposures. And so when we have a subsequent exposure for a secondary immune response here, instead of having the one proliferating into a handful, we'll now have a subset of cells that are there that can have a much more rapid response that can help protect from that subsequent exposure, particularly if it's a dangerous pathogen. And so the ability to develop these memory cells as like one flavor of our T cells and B cells helps promote an enhanced secondary immune response, a response to secondary exposure to this antigen that we've seen before. Yeah.
Do memory cells for T cells proliferate in the same way that cells do?
Mm-hmm. Yeah, and so I should say the response that you've experienced in the lymph node where it feels really tender, it's both the B cell that sees that free antigen that's responding to and the T cell that has seen it through antigen presentation that are both expanding. So you can have antibodies produced by the B cells and T cells that can help mediate the cell-mediated response to attack the pathogen. And so they're both undergoing this proliferation, and then they'll drain out of the lymph node to where the site of infection or inflammation is.
And do those memory T cells fall under a specific category or cells, or is that?
No, that's a great question. So this is an important distinction. Memory cells are not effector cells. Memory cells are these long-lasting that we all have in our circulation right now that are waiting to see their antigen. But it's when the antigen is present that our cells, those lymphocytes, become effector cells. Thank you. That's a really great question. Thank you for that. All right. So taking this all the way back to the beginning of last class, we talked about autoimmune disease, and we talked about how autoimmune disease is our immune response, again, is the big picture function of our immune response is to distinguish self versus foreign. Autoimmunity is when the self is identified as foreign and subsequently attacked, or at least parts of self is identified as foreign. One example is celiac disease. I'm sure a lot of us know people or have interacted with people that suffer from celiac disease. And this is oftentimes an autoimmune attack on our intestinal wall, our GI tract, that's exacerbated by the presence of gluten. Type 1 diabetes is an autoimmune attack on our pancreatic beta cells, where there's a factor on the beta cells that's identified as foreign and are subsequently destroyed. MS, multiple sclerosis, is a progressive neurodegenerative disease that's a autoimmune disease of the central nervous system. And finally, rheumatoid arthritis is a systemic disease that affects our joints, where collagen, that main, we talked about this back during the extracellular matrix, it's a key component of our connective tissue. That's what's typically identified as the foreign substance, which is problematic given that collagen is literally everywhere throughout our body, which is why RA is seen as a systemic disease. What's really important to consider is the etiology of some of these autoimmune diseases. And so one possible etiology for some of them, and that's going to play a role in Guillain-Barré syndrome that we talk about today, is that something called molecular mimicry, where one molecule mimics or resembles another that triggers this autoimmune response. So again, we have an antigen that typically initiates an immune response. What can end up happening during autoimmunity for molecular mimicry is that the antigen receptors on our lymphocytes will cross-react. That means react with something that's not their antigen. Our antigen receptors will typically bind to a specific peptide that's on a specific antigen, but if they cross-react, they'll bind to something else that resembles or mimics our own self in this case, and that own self antigen is called the autoantigen. So we have a pathogen that has a specific epitope on it, and we have an autoantigen that resembles that epitope on some sort of pathogen. And so they mimic one another, particularly whatever this pathogen is, mimics something that's within us and and promotes the autoimmunity, where something in us that resembles it promotes this autoimmunity. And so the downstream consequence of that is that there's that component of our own body that is now identified as foreign and is now attacked by our immune system. And so either that specific protein or that autoantigen might be inhibited, or the cells that express it on the surface will be negatively affected, is one way to say it, or destroyed is another way to put it. So taking this all the way back to Guillain-Barré syndrome, or GBS, we talked about how the cases begin a couple of weeks after infection. And so the C. jejuni is what happens is that it promotes mimicry. There's a biomimicry that happens between C. jejuni and a component within us, within our peripheral nervous system. And so the key things to consider here, like what is the antigen, how is it presented? Think about, is it humoral cell mediated, and what is the autoantigen here? And so these are things to consider as we talk about the pathogenesis and the etiology of GBS. Bless you. As I've cited a few times now, we are focusing on the foundational elements of immunobiology of our immune systems. And so everything we talk about, there's a layer of complexity underneath that can sometimes complicate things. So we talked about MHC class 2 is what is usually displaying peptides from other pathogens. There's also another antigen presentation structure called CD1. MHCs will typically display peptides. CD1 will display lipids typically. And so that's what's depicted here. And what ends up happening in GBS is that there's exposure to C. jejuni in the GI tract, will then cross the intestinal epithelium. This B cell that binds to and recognizes an epitope on C. jejuni will endocytose it, break it down, and display it on its surface. What ends up happening that's really key here is that what's displayed is the antigen, but it's going to resemble something in our peripheral nervous system. And so when it displays and finds a T helper cell, what it's displaying on CD1 is called a lipooligosaccharide. It's a lipid with a lot of sugar molecules on it, basically. And so once that happens, they'll activate one another, and the B cell will differentiate into a plasma cell and secrete antibodies against or anti. Whenever you see anti something, that means antibodies against that recognize lipooligosaccharides. And so what's key to note here is that these lipooligosaccharides on C. jejuni that have been identified resemble a key part of our Schwann cells here. And so the autoantigens here that are key to note here are called gangliosides that are part of lipooligosaccharide. And so they the structure here is nearly identical to what is found on C. jejuni. But these gangliosides are the key mimicry part here. And so, excuse me. And so the autoantigen the autoantigen of gangliosides on our cell membranes are key for cell signaling, which again is key for nerve conduction in our peripheral nervous system. And so when these autoantibodies or these antibodies against lipooligosaccharides are developed, they're going to eventually encounter and attack our Schwann cells that display gangliosides on their surface. The gangliosides resemble a key part of the lipooligosaccharide. And so it's going to attack our Schwann cells that display these gangliosides. That's going to cause demyelination and eventual neurodegeneration. Some of the key treatments for GBS, because it can affect our respiratory system, there's a key need for respiratory support right away. And the respiratory support come in a few different flavors, basically not too different from an iron lung and what we saw in the past. And there's going to be immunosuppression therapy because, again, this is an autoimmune disease. One of the flavors of this autoimmune therapy can look like trying to remove the antibodies via a process called plasmapheresis, which is basically removing the plasma, a key liquid component of our blood, to try and remove some of these antibodies that are at the heart of the disease. And also getting injections of antibodies that will then target these antibodies that target gangliosides, so that there are antibodies that target these other antibodies to eliminate them and neutralize them. If adaptive immunology or autoimmunity is something that you've really appreciated in the last couple of lectures, we have a great cohort of faculty here just in MR6 that way, in the Cardioimmunology Center we actually benefit from having some of the biggest names in adaptive immunology in the country here. We just actually lost a big figure named Tom Bracchiali, who is the main T cell guy when it comes to flu. So UVA used to be a really big center for adaptive immunity and still is. And so with that, thanks for your time, and I'll be hanging back for questions.